Okay, so when a mommy and a daddy love each other very much, fertilization takes place. Fertilization is when a male sperm cell meets a female egg cell. Both of these cells are known as haploid cells. Haploid cells contain half the amount of chromosomes, which is 23, that are contained in a normal diploid cell. Meiosis 1. Once fertilization takes place, interphase starts. Interphase serves as a resting place between cell divisions, and it's also when the cell spends most of its lifetime. During interphase, a cell is duplicating chromosomes and DNA strands inside the nucleus. In terms of meiosis specifically, both male and female chromosomes are being copied. Once interphase is done and all chromosomes are duplicated, prophase 1 takes place. Prophase 1 is when centrosomes, protein cylinders that were next to the nucleus, go to separate corners of the cell. When this happens, it tangles up all the chromosomes and crossing over occurs. Crossing over is when an egg and sperm cell chromosomes cross over together, as you may have guessed, and get attached to its duplicate copy from the opposite gender to form an X-shaped double chromosome. After this, the resulting chromosome will have 4 chromatids. Chromatids are the other half of an X-shaped chromosome, so when crossover happens and they attach to each other, they are actually exchanging DNA. After crossover happens in this stage, they go into the next stage, which is metaphase 1. In metaphase 1, the updated chromosomes align along the middle of the cell, which then brings us to anaphase 1. In anaphase 1, the chromosomes are pulled apart by the microtubules. After that happens, the cell goes into telophase 1. In telophase 1, all the chromosomes gather to each side of the cell, which then brings us to cytokinesis, where the cytoplasm divides into 2 daughter cells. Meiosis 2. The starting phase for meiosis 2 is prophase 2. In prophase 2, a new spindle forms around the chromosomes, which leads to metaphase 2. In metaphase 2, chromosomes line up in the middle of the cell just as before, and they start anaphase 2 when they are divided once more into 4 chromatids. As a result, telophase 2 and cytokinesis happens yet again, when the chromosomes are then pulled apart to the poles of the cell and the cell divides at the cytoplasm, with 1 chromatid in each new cell.